So protein synthesis Labster's about two different objectives. One is to help make a drug to treat anemia, and this is specifically chronic kidney failure patients tend to have certain cells that produce the hormone called erythropoietin stop working, and erythropoietin is needed to produce red blood cells. So then if you don't make enough red blood cells, then you can become anemic, right? Because anemia is not having enough red blood cells, not having enough iron in your blood typically. So a lot of people are used to thinking of anemia as being an iron or at least caused by an iron deficiency. But in this case, you're looking at people with anemia that's caused from not having enough erythropoietin due to kidney disease. So the goal is to make the hormone erythropoietin so that you can give that to the patient, and then it will stimulate red blood cell production. And so you're going to compare RH-EPO, which stands for recombinant human erythropoietin, and you're going to have that produced by both, you're going to have it produced in bacteria and compare the product to mammalian cells, and specifically that'll be Chinese hamster ovary cells, and that's abbreviated CHO. I think that's the mammalian cells that you're using. Okay, so then you're going to compare those two products, the EPO from bacteria and the RH-EPO from mammalian cells. And you'll look at mass spec reading, and you'll compare those that way. And you're also going to do a review of DNA transcription and translation, and you're going to compare what you know of DNA transcription and translation, also called protein synthesis. You're going to apply that knowledge in the making and evaluating the RH-EPO. And then lastly, you're going to help, I don't remember what this person's name is, but you're going to help find athletes that are using RH-EPO to gain an advantage. So the idea is if some athletes think that if they take EPO, then they will have a higher capacity for oxygen carrying, right? They're going to have a higher oxygen load in the blood, which is going to allow their muscles to do a little bit more work. And this is very minor because your kidneys check for the amount of red blood cells and balance this. So the overall effect might only be 1 to 3 seconds, but if you're running a race or playing some other competitive sport, 1 to 3 seconds could be enough for you to win. So so that's why some athletes will do this. And so it's called blood doping, is to get that competitive advantage. And of course that's not allowed. So they will do different types of tox screens on athletes from time to time. And so you'll be looking at urine samples. So in order to see if a urine sample is normal or not, you're going to compare your mass spec readings of EPO, different natural human EPO, and the recombinant human EPO, and then also looking at urine samples of what you know is a normal, right, your control, looking at a normal urine sample that doesn't have any EPO in it. And then you'll compare 3 athlete samples. Okay, so I'm not going to review mass spec with you guys. You can read about that and watch the little video in the Labster about that. So you do need to know it, but I didn't include it in these slides. I wanted to go ahead and use this time to review protein synthesis with you, which is a little bit more involved than the mass spec details. Okay, so super simplified overview of protein synthesis. If this box represents a cell, this oval represents the nucleus, or the nuclear envelope rather, and the inside is the nucleus. And then these squiggly lines are the DNA. And so if you were to transcribe the DNA, you would be making messenger RNA. So here's our messenger RNA. And then you know that messenger RNA will then have to leave the nucleus and find a ribosome. And so here you have the large and small subunit of the ribosome, the mRNA sandwiched in between it, and then these hollow ovals represent individual amino acids. And as you link amino acids together, you have a growing polypeptide. So this is the most basic overview of protein synthesis, and we're going to continue to add on more and more detail to this. Okay, so transcription, and of course, the process of taking mRNA and making protein is called translation. So transcript is taking a gene from DNA and changing it from DNA to RNA, but both DNA and RNA are said to be nucleic acid. So it's the same language of nucleic acid, but the difference from DNA to RNA, of course, you know that instead of T, you'd have a U for uracil. Okay, and then the sugar itself for DNA is deoxyribose, and for RNA it's just ribose. Okay, but they are still both nucleic acids, so we'll say that it's the same language. But then here in translation, you need to translate from mRNA, which is a nucleic acid, and you're going to translate that into a completely different language, which is amino acids, to make a protein, okay? So if you feel like it's hard for you to remember the difference between transcription and translation, you can remember alphabetically, like C, transcription comes first, but you could also remember conceptually that translation's translating between nucleic acid and amino acid language. Okay, so a little bit more detail. In your DNA, you're going to have two strands, right? Because you know it's a double helix. And so on one strand, it's going to be 3' to 5', and on the other strand, it's going to be 5' to 3'. So they go in opposite directions. And only one of those strands will be used to generate your mRNA. And then so mRNA is single-stranded, right? Whereas DNA is double-stranded. Then you take your single-strand mRNA, go into the cytoplasm, find a ribosome, and then the transfer RNAs, tRNA, is going to bring in corresponding amino acids. So every 3 base pairs on the RNA sequence makes up a codon. And so each codon is going to code for a specific amino acid. So in this case, UGG codes for tryptophan, TRP, abbreviated. UUU codes for PHE, which I think is phenylene, if I remember correctly. It's been years since I took biochemistry and I'm a little rusty. And then GGC is for glycine and UCA is for serine. This could be phenylalanine, PAG, phenylalanine. That's probably what it is. Okay. Now over on this side, I'm helping you to see what do we mean by 5' and 3' ends. So here we would say this is the 3' end, and this is the 5' end. But in this picture, the only way to know is because we were told it was labeled. But in actuality, if you could see the molecule, you would see that you start at oxygen and you count your carbons. So first carbon, second carbon, third carbon, fourth carbon, and the fifth carbon hangs out above the ring, and then you have your phosphate group. And so the 5' end is always the end of the strand of DNA that has a phosphate group, a free phosphate group. And the 3' end is the third carbon that has the free, it would actually have an OH, which you can see right here in this picture, OH. In this picture, we've gotten rid of the H and we're ready to make the bond of the next nucleotide coming in and attaching right there. But when you are talking about ends, 5' end and 3' end, you're looking for the 5' end is going to have the free phosphate group, and the 3' end is going to have the free OH group. Okay, and of course it'll be flipped around for the opposite side, right? And OH, phosphate. Okay, so next, some little reminders about transcription, and then we'll go into the steps. Okay, so let me move that out of the way. So the DNA template has a region called the promoter, and that is where the RNA polymerase will bind to the DNA to begin transcription. The promoter itself is not copied, so RNA does not have a promoter, only DNA has a promoter. And as I mentioned earlier, RNA will have a U for uracil instead of the T that you have in DNA. RNA is also less stable, and another way to say that is that it's more reactive. And that is also in your simulation. The DNA template is read by the polymerase 3 to 5 direction, which means that the mRNA itself will be synthesized 5 to 3' direction. And that is demonstrated here. So this would be the template strand, and it's being read 3' to 5'. And as the polymerase goes down the template strand towards the 5' end, it's adding in complementary base pairs to synthesize the RNA, and that RNA is going to be 5' to 3'. And that big oval is the RNA polymerase that's being shown here. So another thing that I might not have said on this slide is that the RNA polymerase also opens up the DNA because it's usually, right, double-stranded, and you wouldn't be able to access the base pairs to copy. So another important first step is RNA binds to the promoter and opens up the DNA strand so that it can copy. Okay, next. So all of the examples I've been showing you are strictly eukaryotic, excuse me, because I've been talking about a nucleus, right? Just want to point that out. So in the nucleus, you have an RNA polymerase that will bind to the promoter. The DNA will unwind, open up, and then the RNA polymerase will make an RNA strand due to adding in the matching base pairs, right? So in a complementary base pair fashion. So remember, A binds to T, C binds to G, and then when it's RNA, which is what we're talking about, if your DNA has an A, instead of the polymerase adding in a T, it adds in a U instead, okay? And it'll keep doing this until it reaches the terminator sequence in the DNA, which will cause it to stop, and the whole complex will fall apart. Okay, and then the mRNA transcript has to get a cap, what we call a poly-A cap, a poly-A tail, and a cap. Sorry, I am definitely tired today. Okay, so mRNA has to mature, and maturation means getting a cap and getting a tail, which will protect it from getting chopped to pieces when it moves into the nucleus, into the cytoplasm. So it has to move out of the nucleus into the cytoplasm. The only way to safely move out of the nucleus into the cytoplasm is to get that cap and poly-A tail, okay? And then once it's in the cytoplasm, it has to find a ribosome. The ribosome could be free floating in the cytoplasm, or it could be bound to ER, right? Rough endoplasmic reticulum. And then you'll begin translation once you have the ribosome. So it turns out that initiation complex actually forms first. So you don't just like the mRNA doesn't just squeeze into a ribosome. Actually, what happens is that the small ribosomal unit, the bottom, the small unit, will bind to the mRNA, and then a transfer RNA with the first codon is always the start codon, so it's going to be a methionine, and it's going to bind, and then the large subunit will bind to that. So it's actually a very specific sequence of events before you have the whole unit. Then you're able to repeat over and over and over again, bringing in new transfer RNAs, specifically matching the codon. So the codon is on the mRNA. The green part in this image is the anticodon, which is just the complement of the codon. And so it's called the anticodon, which is on the tRNA. And on the other end of the tRNA will be the amino acid. And so it always arrives, so this would be EPA site. So your new tRNA comes into the P site, and then as it moves over to the next site, it makes a peptide bond to add on this new amino acid to the growing chain. And then once that's finished, the empty tRNA moves into the next spot, which is the E site. And I always think E for ejection site. And then the whole thing will just continue until you reach the end of your mRNA. So as I mentioned, you'll read the ribosome reads the second codon, which means another tRNA will come in with the matching anticodon and the specific amino acid that anticodon codes for. And then the peptide bond will be made between amino acids, and then it scoots over, and then you repeat until you get to the end. So process repeats until you reach a stop codon, and that's going to cause a tRNA called a release factor to go into the ribosome. And so it goes in, so the stop codon here, the special stop tRNA goes here. Notice it will not have any amino acid attached to it. And that is going to jam up this whole complex. Everything will then be ejected and released and fall apart. Okay, you can have lots of ribosomes on one mRNA. So just because this one falls apart doesn't mean that this mRNA is done, right? So as soon as one ribosome starts, another one's going to start, and another one's going to start, and another one's going to start. So you're going to have lots of protein made very, very quickly. Okay, so let's go ahead and see this in action. So let me move over. And this shows transcription and translation. So let's go ahead and watch this now.
variety of nutrients we get. In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are three distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately.
Okay.
After dissociation,
So that is transcription and translation. Hopefully that was all review for you. If not, study it real hard because you're expected to know all of that already. Okay, I included another video on translation for you. So it's only the translation portion and it's only 3 minutes long. And I'm not going to show that now because we just watched it in this video, but this is another video so you can see it in a slightly different way. It's pretty, it's consistent, it's the same thing, but it's a different animator. So different sketches, drawings. Okay, so let's move on with our introduction. Okay, so after you study, you should be able to tell me exactly what's happening during transcription. Like, what do you need, right? You need your RNA polymerase to bind to the promoter region to unwind, open up the DNA, and then it's going to copy those base pairs in a complementary base pairing fashion, where A goes with a U, and a T will, the RNA polymerase will put in an A. If there's a C in the DNA, then you will add a G, so on and so forth. And the direction that it's going, I think that's pretty much what I told you for transcription. And that produces your mRNA transcript. Know the difference between DNA and RNA. And then lastly, that the RNA transcript has to be matured. So it needs the cap and it needs the tail. Then it will go out of the nucleus and you will form your initiation complex. Know that all the details that I told you for translation, right? You need the small ribosomal unit to bind to the mRNA, and then you need that start codon, methionine, to bind to the first codon, and then the large subunit can bind, and then you'll continue to go down the mRNA and add on corresponding amino acids. And remember that depending on the anticodon, so the anticodon is going to have a specific amino acid attached to that tRNA, and then you're going to grow a long chain called a polypeptide. After that, you know that in order for it to become a functional protein, it's going to need to have all the folding done, but we're not going to look into what we would call post-translation steps. Once you have the polypeptide, as far as this class is concerned, you're done. Okay,